When peripheral stimuli like lipopolysaccharides or cytokines, specifically IL-1, IL-6, and TNF-alpha, breach cellular defenses, they trigger an immediate, aggressive inflammatory response. These localized inflammatory signals travel directly to the brain, reaching the hypothalamus to stimulate the release of corticotropin-releasing hormone, or CRH. This linear flowchart maps the standard biological response. The CRH prompts the pituitary gland to produce and release adrenocorticotropin hormone, or ACTH. That ACTH then travels to the adrenal cortex to stimulate the production of cortisol. This specific sequence is the traditionally understood endpoint of the axis. Standard physiological models focus on ACTH as the signaling link required for cortisol production. Clinical practice presents a paradox that breaks this linear model. When physicians use ACTH gel to treat severe multiple sclerosis relapses or nephrotic syndrome, the patients experience specific, measurable clinical improvements. If ACTH served solely as a precursor to cortisol, prescribing exogenous cortisol would yield the exact same therapeutic profile, but it does not. Cortisol alone fails to replicate the specific efficacy of ACTH treatments. ACTH possesses intrinsic cortisol-independent anti-inflammatory activity, functioning through a redundant tripartite regulatory network. This structural map illustrates the complete network. The first parallel pathway flowing outward from the central ACTH node is the adrenal arm. Here, the intact ACTH molecule travels to the adrenal cortex, binding strictly to the melanocortin type 2 receptor, or MC2R. The immediate output of this specific MC2R binding event is a systemic surge in glucocorticoids, primarily cortisol, released into the bloodstream. Cortisol then enters cells across the body, moves into their nuclei, and executes a genomic suppression of inflammatory transcription factors. This genomic suppression is highly effective, but because it relies on altering gene expression and synthesizing new proteins, it operates on a slower timeframe. The slow, sustained kinetics of the adrenal arm leave a temporal gap. In the minutes and hours before genomic suppression takes effect, the body requires immediate local control over the immune response. To achieve immediate control, the body utilizes a second parallel route, a direct, non-classical immune pathway. This pathway relies on a biochemical transformation. Instead of remaining intact, the long ACTH molecular chain undergoes a targeted cleavage, breaking apart into smaller fragments. This cleavage generates new regulatory molecules, specifically alpha-melanocyte stimulating hormone, or alpha-MSH. This process creates distinct active fragments, beginning with the longer alpha-MSH 1-19 chain. But the secondary fragment, alpha-MSH 1-13, is the most highly bioactive variant for immune regulation. By undergoing this cleavage, the systemic endocrine hormone supplies the specific fragments required for targeted, local melanocortin regulation. Both the intact ACTH molecules avoiding the adrenal glands and these newly cleaved alpha-MSH fragments deploy directly to peripheral immune cells. This cellular schematic tracks their arrival at macrophages and lymphocytes. The molecules bypass cortisol mechanics to lock into specific surface receptors, MC1R, MC3R, and MC5R. The instant binding occurs, it triggers the immediate downregulation and suppression of NF-kappa B activation inside the cell. Without active NF-kappa B signaling, the immune cell experiences a sharp, precipitous drop in the production of pro-inflammatory cytokines. A secondary effect immediately follows. The cell decreases its expression of adhesion molecules, limiting the aggregation of immune cells. The pathway then shifts into an active upregulation phase, inducing the cell to increase its output of the anti-inflammatory cytokine IL-10. Direct melanocortin binding achieves rapid, targeted transcriptional inhibition of inflammatory genes at the site of the immune response, acting independently of cortisol. The third and final pathway moves away from local tissue environments and routes directly to the central nervous system. Inside the brain tissue, ACTH and alpha-MSH find a different set of targets, binding to the MC3R and MC4R receptors. This binding within the central nervous system alters the brain's autonomic nervous output. This shift in output triggers a specific physiological mechanism known as the central neurogenic anti-inflammatory reflex. The reflex utilizes neural pathways to transmit inhibitory signals across the body, reducing systemic inflammation. This top-down neural circuitry delivers an immediate dampening of the immune response, complementing the slower adrenal and localized cellular arms. This complete map shows what occurs during an inflammatory event. A single trigger deploys all three pathways simultaneously from the ACTH node. The adrenal arm builds a slow, systemic genomic defense, while the direct immune and neural arms provide rapid, targeted control over specific cells and reflex networks. Isolating these direct melanocortin pathways clarifies the mechanisms of local immunity, but it does not negate the power of the endocrine system. This balancing scale illustrates the final dynamic. Cortisol remains the dominant force for sweeping systemic immunosuppression across the human body. The MCR pathways provide modulatory control over local immunity in the locations and timeframes where cortisol is either too slow to act or absent. This multi-layered redundancy explains the clinical paradox. ACTH analogs effectively treat complex neuroimmune diseases like multiple sclerosis because they activate the localized and neural pathways that exogenous cortisol alone does not. The integration of the melanocortin system ensures robust immune control, establishing ACTH as an intrinsic regulator of human inflammation.